Hey guys, welcome and welcome back to my channel. This is Dr. Prasanjit Das, Academic JR1 in the Department of Pharmacology, AIMS Kalyani. And this is my first PG seminar. So without wasting any time further, let's get into today's topic. So my first PG seminar is on the topic that is Nobel Laureates and landmarks in the field of pharmacology. So before we go into the Nobel Laureates and the landmarks in the field of pharmacology, let us first discuss about the founders of pharmacology. So the founders of pharmacology are considered to be Rudolf Buchim and his student, that is Oswald Smithberg. So in this, Rudolf Buchim is also considered as the father of pharmacology. And why so? We'll get to know in the next slide. So Rudolf Buchim, he turned pharmacology from an empirical study of medicine into an independent medical discipline. That is by introducing two principles, that is classification of drugs into a natural system based on their mechanism of action, mode of action, and the different effects which are produced by the drugs. And he was also responsible for finding out several pharmacological laboratories and training other individuals and exploring the field of experimental pharmacology. These are the two reasons why Rudolf Buchim is considered as the father of pharmacology, because he converted the empirical study of medicine into an independent medical discipline, that is pharmacology. Now, coming to the father of Indian pharmacology, that is Ramnath Chopra. So, why is he considered as the father of Indian pharmacology? Because of the following contributions. He was the first professor of pharmacology at the newly established Calcutta School of Tropical Medicine. His aim was to make Indian pharmacology a self-supporting one, that is by enabling India to utilize the locally produced drugs. So he wanted to make the Indian pharmacology self-supporting by utilizing the locally produced drugs, and he compiled the first Indian pharmacopoeia. So pharmacopoeia is basically an official publication containing a list of medicinal drugs with their effects and the directions for their use in the year 1955. And this was comprising of many indigenous drugs, and he also developed Chopra's antimony test that was used for the diagnosis of leishmaniasis. And he had a key role in the establishment of CDRI, that is the Central Drug Research Institute in Lucknow, Uttar Pradesh. Now coming to the topic proper, that is the Nobel Laureates in the field of pharmacology. So the first group of Nobel Laureates includes James P. Allison and Tasuku Honjo. So they did the discovery of cancer therapy by inhibition of negative immune regulation. In the year 2018, they got the Nobel Prize for this discovery. So James P. Allison studied a known protein that functions as a brake on the immune system, and so releasing it would lead to the activation of our immune cells attacking the tumor cells. So similarly, Tasuku Honjo found another protein which also acted as a brake on the immune system, but by a different mechanism of action. So these two brakes were later found out to be as CTLA4 and PD1 receptors, as you can see in this image, that is CTLA4 and PD1 receptors. So once the brake is unleashed, the immune cells will attack the cancer cells, and that will lead to the production of the anti-cancer effect. So ipilimumab, that is the CTLA4 inhibitor, and nivolumab, pembrolizumab, that is PD1 inhibitors, are used for the management of cancers like melanoma. So this was about the negative immune regulation, that is inhibition of the negative immune regulation for the anti-cancer effect. Now coming to the next group of Nobel Laureates, that is William C. Campbell, Satoshi Omura, and UU2. So William C. Campbell and Satoshi Omura, they got the Nobel Prize for discovering a novel therapy against the infections which are caused by the roundworm parasites in the year 2015, and along with them UU2 also got the Nobel Prize in the year 2015 for the discovery of the novel therapy against malaria. Okay, so one was against malaria and the other one was against the roundworm parasites. So we'll see the details. That is Omira, he isolated some new strains of streptomyces. So basically this is a bacteria which produce streptomycin, and then he cultured them in the lab. And Campbell acquired Omira's cultures, that is the culture of the streptomyces which was producing the streptomycin, and found that one component of the culture had antiparasitic effect in the infected farm animals, and later on this component was found to be ivermectin, and nowadays we use a more modified version of this, that is ivermectin. So ivermectin's better form is ivermectin. It is used in the management of onchocerciasis and scabies. So here you can see the itchy lesions of scabies and also the cornea being affected in onchocerciasis. So these are the conditions where we are using ivermectin, which was being isolated by the streptomyces bacteria which was being cultured. Now about UU2. So UU2 isolated artemisinin from the plant Artemisia annua. Okay, and it was highly effective against the malarial parasite, that is it killed the malarial parasites at a very early stage of their development. So basically an anti-malarial drug, artemisinin. The next group of Nobel laureates include Alfred G. Gilman, that is Alfred Goodman Gilman, and then we have Martin Rodbell. So they got the Nobel Prize in the year 1994 for the discovery of G proteins and the role of these proteins in signal transduction in cells. So G proteins, that is G protein coupled receptors. So they were the one who did the discovery of the G protein coupled receptors and the G proteins. So a little bit of explanation regarding this. So the cells actually communicate with each other by means of hormones and several other signal substances. So they act on the G protein coupled receptor. So here you can see this is the G protein coupled receptor. So it is actually having three different units, that is alpha, beta and gamma units, and also it is traversing the membrane 7 times, that is also known as that is the reason why it is also known as serpentine receptor. So G protein coupled receptor, when activated, they will activate the amplifier system in the cell. Okay, and this will lead to the production of the second messenger, which will further mediate the effect. As you can see here, the second messenger is being formed here, and this will be converting the inactive enzyme into the active form, and as a result it will mediate the effects of the hormones or the signaling substance which acts on the G protein coupled receptor. So Rodbell showed in 1971 that the transduction of a message from the exterior of the cell, that is from the hormones or the signaling molecules to the interior, requires cooperation of three functional units. That includes a discriminator, that is the receptor that recognizes the different extracellular signals, that is the first messenger. So the first messenger are the different extracellular signals, and then a transducer that requires GTP, that is nothing but the G protein, and an amplifier that generates the large quantities of second messenger, which are responsible for converting inactive enzyme into the active enzyme and hence mediating the physiological effect of the signaling molecules. So we need one discriminator, one transducer and one amplifier for the message to be transferred from the exterior of the cell into interior of the cell, and hence producing a physiological effect. So this was being demonstrated by Rodbell. And Gilman tried to find the chemical nature of Rodbell's transducer. So here you can see one pictorial representation of the G proteins. So in a normal leukemia cell, there is presence of receptor, there is also presence of an amplifier and also a G protein, and this is the reason, because all the three are present, so we are getting the biological effect here. Whereas in mutated leukemia cell, we only have the receptor and the amplifier, but there is no connecting G protein here, so there was no biological effect in the mutated leukemia cell. So what Gilman did was he introduced a purified G protein into the mutated cell, and as a result now the biological effect was present because there is a connection between the receptor and amplifier via the G protein which is being introduced into it from outside. So this is the reason why now you can see the biological effect. So it has confirmed that the transducer, that is the G protein, is required for the connection between the receptor and the amplifier and to get the biological effect, which was not seen in case of the mutated leukemia cell where there is absence of the G protein, that is the transducer. So these G proteins can be stimulatory or it can be inhibitory also, that is GS subtype or it can be GI subtype. So it is a totally different topic which is not a part of this seminar. So we can discuss this on some other day. Now coming to the next group of Nobel laureates, that is Sir James W. Black, Gertrude B. Ilion and George H. Hitchings. So they got the Nobel Prize in the year 1988 for their discovery of important principles for drug treatment. So what was the discovery which was done by Black? So Black's discovery of the receptor selective drugs. So Raymond Alquist suggested already in 1948 that opposite effects of catecholamines were mediated by different receptors in the target organs, which we called alpha and beta receptors. Then Black and his co-workers succeeded in developing the first clinically useful beta receptor antagonist, that is pronethalol, in the year 1962 and propranolol in the year 1964. These decrease the oxygen demand of the heart by blocking the beta receptors, and as a result there will be decrease functioning of the heart. So this will lead to decrease in the oxygen demand. So this was the discovery which was done by Black, and Black also suggested the occurrence of two different types of histamine receptors, that is H1 receptor and H2 receptor, and there were no antagonists available at that time for the H2 receptor mediated effects. Meteamide was found to heal peptic ulcer, but it produced agranulocytosis. So subsequently in the year 1975, Black succeeded in developing cimetidine, which had a marked effect in the treatment of peptic ulcer by the H2 antagonism method, but it did not produce the side effect which was being produced by meteamide, that is agranulocytosis. So this led to the discovery of cimetidine and the H1 and H2 receptor blockers. So this was the contribution of Black. Now the studies of the nucleic acid synthesis were responsible for the discovery of several new drugs, and this was being done by Ilion and Hitchings. So Ilion and Hitchings, they studied the growth of Lactobacillus casei, which is a bacteria, and they found that it was dependent on folic acid or a combination of purines and pyrimidines. So the purpose of the study was to characterize the metabolic pathways which are involved in the biosynthesis of the nucleic acids and to identify the anti-metabolites in the nucleic acid metabolism. And it was found that a base, sugar and a phosphate is the major requirement for the nucleic acid synthesis. So their structure analog can block the nucleic acid synthesis because we are altering this. So if we are having a structure analog of either the base or the sugar or the phosphate, then it will inhibit the nucleic acid synthesis because we will have a defective nucleic acid, and as a result there will be defective function of that particular cell in which that defective nucleic acid is present. So this was being discovered by Ilion and Hitchings. So thioguanine, that is in the year 1950, and 6-mercaptopurine, that is 1951, they finally came into picture. That is basically they inhibited the formation of the regular nucleic acid, and then 6-mercaptopurine was further modified to a form which is called as azathioprine in the year 1957. Now coming to the next group of Nobel laureates, that is Alexander Fleming, Howard Walter Florey and Ernst Boris Chain. So they were given the Nobel Prize in the year 1945 for the discovery of penicillin, which is a groundbreaking discovery in the field of antibiotics. So the penicillin discovery was considered to be an accidental discovery. So the discovery of penicillin is considered as an accidental discovery. This is because Dr. Alexander Fleming, he was doing some research on the Staphylococcus bacteria. So he left several petri dishes which were being cultured with Staphylococcus, and he went for a holiday, and following that when he came back, he found that a particular mold, that is the Penicillium notatum colony, started growing in that culture. That is in the petri dish, and he found that the Staphylococcus colonies which were present close to the Penicillium notatum had undergone lysis, whereas the colonies which were far away from the Penicillium notatum colony, that is the Staphylococcus which are present away from the Penicillium notatum, they were normal and they didn't undergo lysis. So it was found that there was some component which was being produced by this Penicillium notatum colony which had led to the lysis of the Staphylococcus, that is, it is having an antibacterial effect. Later, Chain and Florey discovered that penicillin was not an enzyme. So this antibacterial effect was produced because of the penicillin which is being produced by the Penicillium notatum. And later on, Chen and Flore discovered that this penicillin was not an enzyme but it is a molecule which is unstable. So they were able to dry freeze the penicillin into a powder which is far much stable than the one which they found earlier. This is the contribution of Chen and Flore. So the discovery was done by Alexander Fleming, and Chen and Flore were able to dry freeze penicillin into a powder form which is more stable as compared to the raw form. So this was about the different Nobel laureates in the field of pharmacology, and the list is a never-ending list. So these were some of the significant individuals who had their impact in the field of pharmacology, and these are my references which I have followed. So with this I would like to end this presentation. So thank you guys, and if you like my content, please do like, share, subscribe, comment, and also press the bell icon to get notified whenever I upload a new video. So with this I would end this presentation. Thank you guys.